All right. My name is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. Welcome to February's webinar, which is an ongoing series presented by ASCCT and the European Society for Toxicology in Vitro. I encourage you to visit each of our organization's websites to learn a little bit more about them and consider becoming a member to help support these activities and our annual meetings. And finally, you can also join our mailing list, which is on the ASCCtalks.org website to get meeting announcements and things like that. The webinar is going to be recorded and it will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenters using the Q&A feature in the toolbar at the bottom of your screen. There will be a Q&A after each presenter finishes. So get those questions in while the presenter is talking, and then we can address them in between each presenter. Other participants can see and vote for the questions that you enter. So if someone has the same question as you, feel free to just vote that up with a thumbs up. And yeah, also, since we have 3 speakers today, make sure to indicate who your question is directed to. That's really helpful. Thank you. We're using closed caption subtitles. If you want to enable them or hide them, you can do so at the bottom of your toolbar where you see live transcript. And then please let us know where you're joining from. Say hello in the chat. We'd love to hear from you. So I have a couple of announcements for you first before we get started. First of all, we have set the dates for the 12th annual ASCCT meeting. It's going to be in the DC area, and it's going to be October 23rd through the 25th. The 23rd and 24th will be the meeting program, and then the 25th will be a set of CE courses, which new this year, we're going to be allowing CE proposal submissions. So if you have an idea for a CE course related to the ASCCT mission, please look out for that. The submissions for those CE proposals and also abstract submission for general meeting abstracts and presentation will open in a few weeks. Also, the Physicians Committee is offering a series of training webinars related to the 2-year carcinogenicity assay. The first one is tomorrow, and then there are 2 more each Thursday after that. You can see a little bit of information here on your screen. And if you go to pcrm.org/neura, you can register for all 3 of them. And finally, the Physicians Committee is hiring, so please go to their website to find out more if you are interested. Next month, we will have a webinar featuring 2 award winners from the European Society for Toxicology in Vitro annual meeting, which took place last November. So I'm excited to hear about those talks. That's March 24th. You can register at our website. And then let me get to today. We have a real treat for you today. We have presentations from 3 scientists who won awards as part of the 2022 annual ASCCT meeting. The first 2 speakers won poster awards for their excellent work. One of them is integrating high-throughput transcriptomics into a tiered framework to prioritize chemicals for toxicity testing by Dr. Jesse Rogers, and the other one is evaluation of per and polyfluoroalkyl substances in vitro toxicity testing for developmental neurotoxicity by Dr. Kelly Carstens. And so let me just tell you a little bit more about those first 2 speakers. Dr. Jesse Rogers is currently an ORISE postdoctoral fellow at the Center of Computational Toxicology and Exposure at the US EPA Office of Research and Development, where he works to integrate high-throughput data streams generated by new approach methodologies for hazard prediction during chemical screening. His research interests include mechanistic interpretation of high-throughput transcriptomics and applications of transcriptomics to water quality testing. Prior to joining the EPA in 2021, Jesse received a Bachelor of Science in Chemistry from UNC Chapel Hill and a PhD in bioengineering from Clemson University. Dr. Kelly Carstens is also with the US EPA Center for computational toxicology and exposure. And she works on applying computational approaches and predictive modeling to integrate multi-dimensional, high-throughput new approach methods for chemical prioritization and risk assessment with particular expertise in the field of developmental neurotoxicology. And then finally, our last presenter is going to be Anne van der Zalm. She's an advisor to the PETA Science Consortium International, an organization promoting robust non-animal testing methods that protect human health and the environment. She receives her master's in chemistry from the University of Oxford in the UK in 2015. And she worked in regulatory affairs prior to joining the consortium, where she now collaborates with regulators and other experts to further the development and implementation of reliable in vitro toxicity testing approaches. And in 2022, Anna won the Ed Carney Predictive Toxicology Award for her work, which she will present today, which is a modern framework to establish scientific confidence in new methods. Okay, and now I will stop sharing and I will give the floor to Jessie.
Thank you. Thanks, Christy. I'll just share my screen here. Okay. Can you see this okay?
Yep, looks good.
All right, awesome. So thanks for having me here today. I'm excited to present some of the work I've been doing during my postdoc at EPA, and I will just dive into it here. So before I dive in, I'll just add a quick disclaimer that the views expressed here are my own and do not necessarily represent the views or policies of the EPA. And any trade names mentioned also don't constitute an endorsement of that product. So the rationale for this work really comes from the current gaps that come from an animal-based toxicity testing paradigm, right? Where one of the many issues within that realm is we have many chemicals in registration and in use in the U.S. and far too little data for a comprehensive assessment. And so documents like the National Research Council's toxicity testing in the 21st century kind of started to herald this shift from animal-based toxicity testing to next generation risk assessments and the use of NAMs for toxicology. So several programs in the past, as we all know, have been developed towards this effort. One being the US EPA's ToxCast program. One component of that, of which is a suite of high throughput screening assays to assess bioactivity of chemicals, right? And this 2016 paper by Richard et al. has kind of summarized the current chemical landscape of the ToxCast program. And so while we do have a number of chemicals for which we've identified and profiled a large number of biological endpoints, the chemical universe extends far beyond the reaches of this graph. And so the overall goal of being able to identify potential targets of concern still remains a challenge. And so particularly it's very resource intensive to fill this gap in screening using high throughput screening assays. And so we kind of have to shift the paradigm a little bit, right, to be able to efficiently screen for hazards with chemical exposure. And so one of the documents that came out of this effort is the CompTox blueprint from the EPA, as we refer to it as, that kind of calls for integrating multiple assay technologies into a single framework. And so that document, which was published in 2019, calls for a tiered hazard evaluation framework. In other words, a framework to really efficiently measure chemicals and their potential hazards by first using some sort of broad profiling assays. A couple of examples being transcriptomics, where we can measure the abundance of various transcripts across, the human transcriptome, and phenotypic profiling, which can measure thousands of morphological features of cells upon chemical exposure. From there, if a chemical, shows some sort of activity in a tier 1 assay, then we would advance it to tier 2, where we would undergo targeted screening for orthogonal validation. And then from there, we would further advance to tier 3 if a chemical shows some sort of bioactivity of concern, either relating it to an existing AOP or not, if there is no existing AOP, and therefore kind of starting to streamline the screening process, when it comes to this large universe of chemicals. And so the question, or one of the questions that pops up is, how do we start to translate across these different tiers of assays, right? And so start to distill down this high dimensional data that we generate in tier 1 into a targeted hypothesis that we can then test in a tier 2 assay. And so so this becomes more of a computational problem, right? In terms of how do we deal with all of this high dimensional data. And specifically, I'll talk about transcriptomics here. And there's certainly an opportunity to be able to distill information about MOAs, or mechanisms of action, from these transcriptomic NAMs. And then further, there's still this need to develop criteria to prioritize chemicals from tier 1 to tier 2, and so on. So that's really becomes the goal of my talk here. I'll start by talking about an exercise in using signatures from transcriptomic data to profile for activity based on particular mechanisms. Then I'll talk about how we insert that into a preliminary tiered framework to prioritize chemicals for those particular mechanisms. I'll talk about applying that framework to retrospective data from our HTTR and ToxCast screening programs. And then if there's time, I'll talk about, doing some prospective identification of candidates based on tier 1 data. So to give a little bit more context to the transcriptomic assay that we're utilizing for the study, this data comes from Josh Harrell's lab here at CCTE. And his group has done a tremendous amount of work in the past several years to develop the infrastructure and the protocol for implementing transcriptomics for large scale chemical screening. So the basis for this assay is the TempoSeq assay, which is measuring rather than individual expressed transcripts from a cell line upon chemical exposure. We're measuring the abundance of probes that have been hybridized to those expressed transcripts to increase efficiency of the assay. And so with that, we can kind of start to scale up, right, to multi-concentration format and for thousands of chemicals. And so we've conducted various screens across 3 different cell lines, the last 2 of which I'll be talking about today. And so in the 2021 paper that came out, our group discusses the protocol that we've established both on the wet lab side with automated chemical dosing and cell culture, but also on the bioinformatics side with folks like Richard Judson and Logan Everett, who have really done the heavy lifting, developing a pipeline for robust and efficient analysis of differentially expressed genes. And the part that I want to focus on here is kind of the end of this pipeline, where for any given chemical exposure at a dose, we can profile those genes and we can measure differential expression using standard software. But then we can combine those using concentration response profiling and ultimately get an estimate of the point of departure, right? And so we can do this both on an individual gene level, looking at the change in the log2 fold change, or we can do it on a signature basis by aggregating genes using gene set enrichment analysis. And so ultimately we can take a concentration response profile like this, fit it to curves using software like TCPL Fit2, and then ultimately estimate a benchmark dose as our point of departure, along with confidence intervals. So we have this sophisticated protocol, right, for deriving potency information, but then the question becomes what kind of mechanisms are of interest, and would be of interest for kind of a tiered framework. And so in our case, we were interested in gene sets not only that show concentration responsiveness, but also are linked with these individual mechanisms of action by their potency. And so this process allowed us to actually derive our own signatures from our existing data and come out with these what we call RCAS, or reference class associated signatures. So this process is predicated on individual reference chemicals that are annotated for a particular target of interest. And so RefChemDB is a resource that has a lot of these annotations by Richard Judson and Nancy Baker and others at our center. And so what we can do is we can look at those specific chemicals that have a common target. We can pull the genes from TempoSeq in our assay, and we can look at the benchmark doses or our potency estimates for each gene. And then we can run a pretty simple series of one-way ANOVAs that test for differences in the benchmark dose as a function of the reference class. In other words, the putative mechanism for those chemicals. So if a gene shows a significant ANOVA result, then we can compare that gene's potency for individual classes. And then ultimately we can assign that gene to one individual class if we see evidence that that gene is more potent for that class compared to most of the other classes. And so what we get out of this at the end is a subset of genes for which we see distinct potency with regard to just a subset of reference chemicals with a common target. And so it kind of starts to distill down this large transcriptional landscape into just a few genes that represent unique mechanisms. So we ran this exercise across 2 different cell lines worth of data, both in HEPA-RG, hepatic liver cells, and also U2OS osteosarcoma cells. And just to give you a sense of what we found here, each heat map representing each cell line compares the genes in the columns to reference chemicals in the rows. And so for these subsets of genes that you can kind of see one example of here, we have distinctly potent gene behavior for only a subset of reference chemicals, like the aryl hydrocarbon receptor agonists here. And if you look at those same genes for a different group of reference chemicals, you see largely little potency and very inconsistent potency across those chemicals. And so this pretty simplistic exercise can show us that we can actually mine this data for unique patterns, not only in expression but also in potency across these chemicals. And so that could potentially be a useful screening tool and translatable one. So we additionally wanted to look at how we can translate this in kind of a screening context. And so rather than looking at individual genes and their expression, we wanted to aggregate these genes into a signature basis. And so for each one of those gene sets that we identified, we ran gene set enrichment analysis for each chemical at each dose. And using that dose series of enrichment scores, we can then estimate the potency on a signature basis using profiling software. And so as an example here, for each one of these panels representing an individual RCAS signature, we compared both the potency on the x-axis and the efficacy on the y-axis for chemicals annotated for that target in blue to the chemicals annotated for some other target in red. And you can kind of hopefully appreciate the differences in both potency and efficacy between these in-class and out-of-class chemicals, which to us kind of supports the idea of using signatures as a basis to distill down this transcriptomic information into more mechanistic predictions. So for kind of the next portion of this talk, we wanted to insert, a strategy such as this for tier 1 screening into kind of a larger framework. Again, comparing these broad profiling assays to something that's more targeted, akin to ToxCast. So we developed these kind of 4 progressive criteria here with the intent of selecting chemicals that are selectively targeting a mechanism of interest. So our first 2 criteria are in our tier 1 assay here, high throughput transcriptomics, in which we screen for one of those RCAS from the previous slides. Is a chemical bioactive for that signature? And then we add in this additional criteria that's essentially asking, is the chemical acting on that particular mechanism? And is that the reason that we're seeing high potency? Or is that chemical just potent across a number of different targets? And so we may not be confident that chemical is actually hitting the target of interest. And so we add this additional selectivity assessment where we derive a non-selective point of departure from the larger transcriptomic data set and use that for comparison. So a chemical that passes both of our tier 1 criteria then advances to tier 2, or our high throughput screening, where we look at receptor level assays in ToxCast annotated for the same target. And then again, we run this kind of selectivity criteria where we compare to a number of different targets in ToxCast. And so ultimately, when we applied this to retrospective data of just over 1,200 chemicals that were screened in HTTR, we were able to distill down the number of potential candidates into only 67, which is a far more digestible number for a more resource-intensive assessment in the future, using something like an organotypic assay. We wanted to compare the association between our tier 1 screening outcomes and our tier 2 screening outcomes, just as a proof of concept for the tier 1 signatures. And so we took those 1,200 chemicals that were screened in HTTR, looked at whether those chemicals were positive or negative for one of our signatures, and then compared that outcome to its outcome in one of the orthogonal endpoints in ToxCast. And so when we took a look at that using odds ratios and Fisher's exact tests, we found that chemicals that showed activity for one of these RCAS were significantly more likely to show activity in the orthogonal ToxCast assay. And so for us, that helped ensure a bit more confidence that this tier 1 screen is actually starting to filter down for chemicals that are more likely to show activity, and thus contributing to more efficient screening. We also wanted to compare these tier 1 signatures to overall points of departure that you would get from either the tier 1 screening data or the tier 2 screening data. And so first off, we compared our RCAS points of departure versus a benchmark dose that we derived from over 10,000 signatures from public databases. And so this signature set covers a broad range of biology, right? And so presumably the most potent activity of that would be some of the more potent transcriptional activity induced by the chemical, right? And so you can kind of see the results here of that comparison at the top here, where we have the target specific benchmark dose versus the overall transcriptional point of departure. And you can largely see a pretty decent alignment of those points of departure, right? With most chemicals being within half a log of their point of departure, or with the RCAS being below that even. And so that kind of goes to show that the RCAS gets at that more potent transcriptional behavior compared to the larger biological space. We also repeated this exercise compared to a ToxCAS point of departure. And largely we saw that the ToxCAS point of departure was actually lower than the transcriptional one. And so this can be for a number of different reasons, right? Whether it's the dose range tested, which often was larger in ToxCAS, the sensitivity of the detection technology, and so forth. But it also suggests that the tier 1 screen may not be sufficient alone to establish a point of departure, and maybe orthogonal assays are needed for that quantitative estimate. I also wanted to show, just as an example, some of the candidates that were advanced through this tiered protocol. And so any of these chemicals to the right here were active in both tier 1 and their orthogonal tier 2 assay. And so for one of these 3 ToxCAS GR assays. And so in addition to our positive reference chemicals in black, we also identified several chemicals where we didn't have a sufficient annotation, but there was evidence either in the literature or as a registered drug or registered ingredient of it being a glucocorticoid. Lastly, we, very quickly, we did do some prospective screening of a few candidates for which we didn't have sufficient tier 2 data. Out of that, we found that a subset of chemicals that were, they showed tier 1 activity based on the signatures, but were inactive in the orthogonal endpoint in green. A smaller subset were active and were supported by that orthogonal endpoint in orange. And so, ultimately, this kind of goes to show that tier 2 screening is very much necessary, right, for ensuring confidence and in chemicals that we would advance further for assessment, and it assists in potentially deprioritizing other chemicals. And then further, the testing of various endpoints that are highly related and have similar transcriptomic profiles in tier 2 can really distinguish between those individual targets, which has pretty substantial implications in terms of their adverse outcome pathway. We also estimated differences in potency values across the 2 technologies and found some differences by their individual target that's profiled. hERG inhibitors tended to have similar potency values across the tiers, whereas GR agonists showed higher potency with regard to the tier 1 screening. And so, overall, while this prospective screen is quite small, and I would caution, larger conclusions to be drawn, it does seem that the sensitivity of these tier 1 screenings may be context dependent. And so we may need additional screening based on further cell lines or, further assessment of reference chemicals as well. So just to sum up here in the last minute or so, this strategy for identifying mechanisms from HTTR data can be quite simplistic, but it can also be quite powerful for distilling down, these broad profiling assays and linking them to confirmatory assays. The next steps for this, there are plenty of opportunities, including the inclusion of additional data streams like morphological features through high throughput phenotypic profiling. There's also a lot more opportunity to provide further validation for this type of analysis. And I'll just close by acknowledging, all of the fantastic researchers at EPA and the center, Katie Paul Friedman and Logan Everett, who have been great mentors. Josh Harrell, whose lab was instrumental in this work, and Richard Judson and Enron and Joe Bundy as well. And I would love to answer any questions that you have. So thank you.
Thanks, Jesse. That was a really nice overview of some of this really cutting edge work. So thanks for that. We have a couple of questions, and I think we're going to do sort of a hybrid Q&A where we'll save some questions for after everyone is done, and we have like a specific question that I might pose to you right now, just because it's specific to your talk. I noticed there's quite a few questions going in the chat. So if you could please put them into the Q&A, it makes it a lot easier for us to keep track of all the questions as they come in. So that's very much appreciated. So for Jesse, there's a question about validation, basically. So transcription data sets are messy, not only due to dimensionality because of unannotated splice variants in many genes. Are the data from TempoSeq validated by some other method like RT-qPCR?
Right. So, I would say yes. There are many other groups who have utilized this assay in the past, and started to validate some of these assays as well. We do see some commonalities with our high throughput screening assays, such as high throughput qPCR that has been conducted as part of ToxCast. So I would recommend them to check out some of the work from others like Health Canada, and NIEHS, who have done a lot of work in utilizing this assay. But yes, there is definitely validation. Validation in terms of this particular work kind of relies on this use of reference chemicals, and that's certainly an area that we hope to improve and further screen in the future.
Great. I'm always up for exploring ways to effectively but quickly and efficiently validate new methods so that we can keep up with science, all the new science that's coming about. Okay, so we have a couple more questions, but I'm going to save them for the group discussion at the end. So as I said, everybody keep putting your questions in the Q&A. Thank you very much. You've got some kudos in the chat, so go check that out, Jesse, and we will move on to Kelly. Would you like to go ahead and start sharing your screen? There you go. Everything looks good, Kelly. Please take the floor.
All right, so I'm an investigator at the US EPA, as Christy mentioned earlier, and I really appreciate the opportunity to share my work with you today. So first, I'm going to start off with conflict of interest. The views expressed in this presentation are mine and do not necessarily reflect the views or policies of the US EPA. All right, so PFAS are structurally diverse synthetic chemicals that have widespread commercial use, industrial applications such as surfactants and surface protectors and firefighting foams. So the core structure of PFAS are per polyfluorinated carbon chains bonded to different functional groups As of 2021, the OECD adopted a broad definition of PFAS that requires a minimum of 1 perfluorinated carbon, which resulted in a list that approaches nearly 40,000 substances that fall under the term PFAS. Due to their unique chemical stability and long half-lives, concerns have been raised about their environmental persistence, their accumulation, and their toxicity potential. And PFAS are considered ubiquitous in the US, with PFOA and PFAS detectable in more than 90% of the population, with exposure through drinking water, air, or diet, just to list a few. And PFAS have been detected in the groundwater, soil, and in wildlife, but only a small number have been extensively evaluated for adverse human health potential. Probably most well-known are PFOA and PFAS listed here, which are currently under regulation internationally. So what is known about PFAS and developmental neurotoxicity potential? So very little is the answer. The positive experimental data evaluating large number of PFAS is a discernible limitation in understanding the human health effects of PFAS in the developing brain. Although there have been a lot of systematic reviews and meta-analyses of human epidemiological studies, the associations between PFAS, and again, this is predominantly PFOA and PFAS, are inconsistent. And this is similar across experimental animal studies. So really precludes the definitive conclusions regarding what the associations are between PFAS and developmental neurotoxicity. It is known, however, that PFAS are found in the brain. A lot of studies have been conducted in our aquatic mammals like whales and in cord blood. So that's really indicating that PFAS can cross the blood-brain barrier and the placental barrier. So given the challenges in the available studies and really interpreting the associations, there's a huge need for alternative approaches to evaluate a large number of PFAS. So today what I'm going to show you is an analysis that demonstrates the power of using both high throughput screening and computational approaches to evaluate a large number of PFAS screened in a multidimensional battery of developmental neurotoxicity new approach methods. So first, a little bit about the selection of the 160 PFAS that were screened across the D&T NAMs at the US EPA. The PFAS were prioritized and selected based on factors such as structural category, solubility in DMSO, and structural diversity that supports read across. So over here, I'm showing you a plot that shows 160 PFAS that were profiled into different OECD chemical structure categories, with the highest representation falling into the other category here. For more information on the structure-based chemical categories, I'll point you to this very recent publication from our center with the first author from Grace Padowitz. And also note that the PFAS were selected prior to and after analytical quality control testing in the lab of Barbara Wetmore. And I'll also point you to this recent publication, first authored by Marcy Smeltz, to learn more about the QC results. But I'll talk a little bit more about that in a later slide. So the 160 PFAS were screened in both single concentration and multi-concentration across 4 D&T NAM assays: the neural network formation assay, neurite outgrowth, proliferation, and apoptosis, using 2 different assay technologies. So the first assay technology is this microelectrode array network formation assay, or MEA-NFA, which is an assay that measures changes in neuronal activity. And this assay we used primary rat cortical neurons that were plated in a 48 well plate, with each well containing 16 electrodes to record changes in neuronal activity from different types of activity listed here. And the data were collected at multiple days in vitro across development, starting at DIV5 and ending at DIV12. Our second technology is a high content imaging technology, which is an automated data acquisition analysis and analysis system for chemical screening that measures a variety of different morphological features of individual neurons. So in this technology, we measured neurite outgrowth, proliferation, and apoptosis. And then once all the data was collected, it was then put through the ToxCast pipeline to perform concentration response modeling, where ultimately we generated estimated potencies. So what did we find? Well, one of the major findings was that the majority of the PFAS were inactive across the DNT NAMs. Out of 160 PFAS, 118 were inactive, leaving 42 active PFAS that decreased some measure of our DNT assay. And looking at the activity overall, the MEA NFA in green here was the most sensitive to perturbations, with 32 out of the 160 demonstrating activity, and the apoptosis assay demonstrating the least amount of activity, with only one PFAS demonstrating a positive response. So what did we find in terms of potency? So here we're looking at the concentration at 50% maximal activity, or our AC50 value in our concentration response curve as our metric of potency. And what we found is that the general activity categories, so that would be, for example, mean firing rate in our MEA NFA, was the most sensitive and or the most potent overall, with a mean AC50 landing around 0.5 log 10 micromolar. So that converts to about 3 micromolar. And you'll notice that the data points indicate whether the activity was considered active in gray or equivocal in red. So there are a couple of endpoints or data points here that are in this more potent area that were considered equivocal, meaning that expert determination of the concentration response curve determined to be low confidence. And then we also have the data points indicating whether the PFAS passed quality control. And so the asterisk data points are PFAS that failed QC. And so that's a lot of these PFAS in here. And I'll talk more about that again later. So next, we evaluated potential bioactivity profiles of active PFAS by integrating across all the endpoints. And out of the 116 PFAS screen and multi-concentration MEA NFA, 24 PFAS demonstrated moderate to low selective activity. So as Jesse talked about, so our selective activity is activity below a threshold of cytotoxicity. And here I'm defining that by an area under the curve up to the threshold of a cytotoxicity AC50 for the respective assays in the DNT battery. So this is a readout of both efficacy and potency. So coming over to our heat map, which has the endpoints along the columns and our different DTX SIDs of different PFAS on the rows, and our different activity types along the column legend, we see that there are 3 distinct clusters that compare to one developmental neurotoxicity reference chemical, tributyl that is down here at the bottom. And you can see that on the scale of the area under the curve, the dark purple is our higher area under the curve. So that's very high selective activity and efficacy. That most of our PFAS are demonstrating very moderate to low activity, with 4 PFAS demonstrating more activity across all endpoints, another cluster where they're moderate to low, across about half of our endpoints, and then another cluster that are demonstrating more spurious activity. And in this heat map, we also explore the hypothesis that longer perfluorinated carbon chains are associated with elevated levels of bioactivity, which has been suggested in the literature. And so this darker gray color is identifying PFAS that contain perfluorinated carbon chains of more than 8, a length of 8 fluorinated carbons. And it does look like most of them are landing up in our higher bioactivity clusters, although the trend isn't super clear necessarily across these different groups, the 3 different groups that I've binned. Next, we compared the activity of PFAS to non-PFAS chemicals that have been screened in the D&T NAMs. So this could include other D&T reference positives and negative chemicals. And in this cumulative density distribution plot, we see that the median potency of PFAS was near the median potency of other chemicals. And that the median PFAS was right shifted from 2 known developmental neurotoxicants. I picked out methylmercury and tributyltin here. And then looking at the same data in a density plot, you can see that there's a lot of overlap in the potencies between PFAS and non-PFAS chemicals, although PFAS chemicals in gray tended to have a lower density at the more potent concentrations here. So these plots are really confirming the finding that PFAS demonstrate moderate to low potency in the D&T NAMs compared to other chemicals tested, as well as a couple known developmental neurotoxicants. Next, we compared PFAS in the D&T NAMs to activity in other NAMs. So this comparison includes 2 assays known as the biomap and AdaGene assays, which measure perturbations in vascular and immune biology and transcription factor activity, respectively. And the main finding shown in this pie chart from this analysis is that PFAS that were active in the D&T NAMs were also active in the biomap and AdaGene assays, as shown in red here. And only about 5% of the PFAS, which is about 8 chemicals, were active in only the D&T NAMs. Next, we compared the potencies for those PFAS that were active in all 3 assays, and found that the D&T NAM potencies were left shifted from the other assays, sort of indicating that they may be more potent overall. However, we also evaluated whether this was a common trend for the D&T NAMs compared to other assays, and did find that other non-PFAS chemicals were also left shifted in the D&T NAM assays compared to the AdaGene and biomap. So we cannot necessarily conclude that the PFAS disrupt neural systems at a more potent concentration compared to other in vitro targets. So this analysis overall revealed that PFAS bioactivity was unlikely to be neural specific, given that very few PFAS were only active in the D&T NAMs, and actually that a majority of PFAS were inactive in the D&T NAMs, but active in other assays. So that's this green chunk of the pie chart. Next, I evaluated the physicochemical properties and structure feature descriptors, and found that the carbon to fluorine ratio and the octanol water partition coefficient, or log P, were increased in our active D&T NAMs. We also evaluated the length of the perfluorinated carbon chains on a continuous scale. So this y-axis is indicating the number of perfluorinated carbons, and found that there was no significant difference. But this was on a continuous scale. So next, I evaluated in more depth some of the structure feature descriptors to see if there maybe more associations going on than was obvious just by looking at a t-test across the perfluorinated carbon chain length. So in order to look at the structure-based chemical categories, we use an initial set of 34 PFAS-specific tox prints that were constructed from Boolean combinations of the public set of 729 chemotype tox prints, which are available on the dashboard. So these images here were generated using the chemotyper software, which is available here. And I pulled out 5 examples of our PFAS-specific tox prints so that you can kind of wrap your head around what is going on in this analysis. And I overlaid these tox prints with 4 of our most active PFAS in the D&T NAMs. So what you can see is this perfoctyl group, which is identifying a carbon chain of length, 8 perfluorinated carbons, is identified in 3 of our most active PFAS. I also, excuse me, I also highlighted the sulfonyl group as well as the acrylate group, so you can understand where these moieties might fall and their attachments to the perfluorinated carbon chain. All right, so, and looking across these 34 PFAS tox prints, what you can see is that there are some PFAS tox prints landing over in the higher activity area. So this is the number of endpoints that were active in the DNT NAMs. And there were also tox prints that were fairly inactive across the DNT NAMs. And what you'll notice is that the sulfonamide and acrylate groups were the highest overall, but that if you look closely, I'm also showing that not only are some of these tox prints active, highly active, but there were also PFAS containing these tox prints that were inactive here. So this is more of a qualitative view of the level of bioactivity by tox print. But in terms of running statistics on this analysis, none of these would necessarily come out as significant. So I also ran a statistical test, a Fisher's exact test, to run what we call our chemotype enrichment analysis. And the only tox print that came out as significant in terms of bioactivity was the carboxylic acid moiety, which is the COOH, that was enriched by significance in active PFAS compared to inactive PFAS in the DNT NAMs. And then lastly, I mentioned a lot of the analytical QC data that was done on this set of PFAS. So this is really an important factor to take into account in the interpretation of the screening bioactivity. So each PFAS was given a pass or a fail based on whether the parent compound was detected in mass spectrometry. And it's important to note that a failure in this QC result to detect a specific analyte does not necessarily mean that a chemical was not present in the stock. It just meant that the parent compound was not detected. So the results of that analysis across the 160 PFAS were that 43 out of the 118 inactive PFAS failed QC testing. And so this is indicating that these samples were probably unstable or not amenable to screening and aqueous assay technologies. One factor could be the temperature in the mass spec machine, that particular chemicals based on physicochemical properties may have volatilized. And then of the 42 active PFAS, 10 of those failed QC testing. And the activity that we see here, we might hypothesize that this activity resulted from uncharacterized metabolites or degradants. And as showing over here, I looked at 4 different physicochemical properties as they relate to whether the PFAS failed or passed. And you can see some pretty clear trends that molecular weight, log P, and the boiling point were decreased in the PFAS that failed, and the log 10 vapor pressure was increased. So, so for the PFAS that did fail quality control, caution is recommended in the interpretation of the bioactivity in these in vitro assays, although this analysis has really helped flag particular PFAS for further analytical assessment. So in conclusion, I showed you that a set of 160 PFAS representing distinct structural categories were largely inactive in the DNT NAMs, and that a small subset of PFAS demonstrate relatively low efficacy and high potency. We found that a majority of the DNT NAM active PFAS were also active in other NAMs, and that very few PFAS were only active in the DNT NAMs. Findings support recent studies that report an association between PFAS containing carboxylic acid or sulfonamide functional groups as elevated toxicity potential. And additional screening, including PFAS representing overlapping diverse functional groups, and analysis of toxicokinetic parameters will be really important for improving the interpretation of the D&T potential posed by these chemicals. So as you could probably appreciate throughout the presentation, this was a large group effort. A lot of the assays were run in the lab of Tim Schaefer. Katie Paul Friedman was critical in supporting the analysis, and all the other members of the lab, as well as people who've analyzed the quality control data. I will also give you a heads up for a future paper that's coming out from Grace Padowitz and Ann Richard from our group that's going to give a more mature insight into some of the structure-based categories. So with that, I like to thank ASCT and the opportunity to present this work, and feel free to reach out to me at this email if you have any further questions. Oh, and last thing is that all of this data is available in ToxCast at these links here. So thank you.
Great, thank you, Kelly. Really nice of the moment example of how we can use these assays to look at chemicals that people are concerned about. So there's quite a few questions. I'm going to hold some of them, but I wanted to ask you a specific one about the assays. So someone's wondering, in terms of the biological activity that the MEA and the NFA assays measure, can you explain the difference between those two?
Between the, here, I'll go back to my assay slide. Between the two assay technologies, is that the question? Or just between the different types of activity in the MEA and NFA?
This is the same assay, MEA, NFA. I think,
okay.
Right? Is that right?
Yeah, so this is an assay technology.
Different,
yeah. Yeah, on the left. So yeah, I'll just expand really quickly on some of the different types of activity that are being collected. So the general activity bin would capture endpoints like mean firing rate or changes in the number of active electrodes within a well. We also look at measures of network connectivity. So that would kind of capture the synchrony across different electrodes, how many neurons are firing or bursting at the same time, which is an indicator of a healthy, developed, network formation over time. And then we also capture endpoints that measure bursting and oscillatory activity. And at the end of the assay, we capture a cytotoxicity measurement. So hopefully that answers the question.
Okay, thank you. So we're going to move on to the third presenter. Kelly, feel free to type some answers to some of these specific questions if you have the time, and
sure.
We'll cover the rest of them in the end Q&A.
Great.
Okay, so next presenter is Anna van der Zalm from the PETA Science Consortium International. So Anna, feel free to take the floor.
Thanks, Christy. Let's hope this works. Okay, so
we're seeing the...
You can see presenter mode.
Yes, go ahead and switch.
Okay, can you see it now properly?
Yes.
Oh, perfect. Okay. So yeah, thank you so much for having me here today to talk about one of the projects that we've been working on within the PETA Science Consortium International. And for those of you who aren't familiar with our organization, we were formed over 10 years ago now in late 2012, and we're made up of now 26 scientists spread throughout the US, UK, Germany, Belgium, France, and India. And we focus specifically on toxicity testing, and in particular on advancing the implementation and regulatory acceptance of reliable and relevant animal-free testing approaches. And we do this via a number of routes. So by funding the development and implementation of test methods, by organizing free training opportunities, hosting free workshops and webinars, conducting data analysis reviews, publishing and presenting on our work, and so forth. And as we're very involved in the space of new methods, we quickly became aware of some of the barriers that are slowing and in some cases halting progress of newer methods altogether. So for example, the current processes used to validate new approaches are costly, time-consuming, and they don't necessarily produce methods that are fit for regulatory purposes. So to break down some of those barriers, we collaborated with regulators and standard-making organizations to discuss ways to establish scientific confidence in new methods and therefore speed up validation, approval, and implementation. And so today, I'll start by discussing the general framework that we proposed in a paper published last year with those collaborators. And then I'll focus in on how to rethink our assessments of relevance and accuracy of those methods. And I'll end with a case study on the assessment of available eye irritation test methods. So our publication from last year, which is shown at the bottom of the slide, describes a robust, flexible framework for establishing scientific confidence in new approaches. And the framework builds upon the principles of OECD Guidance Document 34 on the validation and international acceptance of new or updated test methods, as well as an evaluation framework specifically supporting NAM assessment for skin sensitization, which was published in 2018 by the International Cooperation on Alternative Test Methods, or ICATM. And so we built upon these and other efforts to develop 5 essential elements which should be considered when assessing new methods for their readiness for regulatory use. And the paper was put together in collaboration with experts from the US Environmental Protection Agency, the European Commission Joint Research Center, the OECD, the US Consumer Product Safety Commission, and the US National Toxicology Program. And as the main figure from the paper highlights, the framework proposes that scientific confidence can be established when these 5 essential elements seen on the outside of the figure of 8 are considered. So for instance, when the information about the model and data are publicly available to the greatest extent possible, when they're reviewed by independent third parties, when the purpose of the model is clearly identified, when the model has been technically characterized, and when the model captures key aspects of human biology or mechanisms of toxicity. And these 5 elements are shown as part of the figure of 8 because the order of assessment may vary. And some can be done in parallel or iteratively. And so now I will go on to describe some of these elements in a bit, well, all of these elements in a bit more detail. So 2 of the overarching elements for gaining confidence are data integrity and transparency and independent review. And as far as data integrity and transparency, there's a need to assess the integrity and credibility of the data from the raw data all the way through to the final reports. There's a need to communicate transparently and as far as possible, make information about the method publicly available. And there's also a need to assess and describe the uncertainties and limitations associated with the method. Independent review is another important part, and for each agency and each method, the appropriate level of external review necessary will need to be determined. So this might range from a peer-reviewed publication of the NAM, detailing its relevance, fitness for purpose, and technical characterization, but it might also be that the method needs to be reviewed by an independent scientific advisory panel, for example. Another element of the framework is fitness for purpose. So determining a method's fitness for purpose involves understanding how, when, and why a method will be used and who will use it. So for example, we should consider which regulatory statutes the data is intended to comply with. So for example, that could be TSCA for the EPA or REACH for ECHA. It also involves defining how the NAM will be used. So will it be used as a standalone assay as part of a defined approach? Will it be used in a weight of evidence assessment? It also includes defining whether the information provided is sufficient to address the regulatory endpoints of interest and if the level of uncertainty is acceptable for the specified purpose. And then finally, it involves defining the context in which the NAM is intended to be used. So is it intended for screening and prioritization? Is it intended for chemical grouping or hazard identification and characterization or quantitative risk assessment? Human biological relevance is the fourth of the 5 essential elements. And simply put, we wanted to highlight with this element the importance of demonstrating similarities between the test system used and human biology. So whether that's the physiology of the test system or the biology measured by the test system used, we want to determine how far the test system reflects human biological understanding. And so this came about because although relevance is a key principle of validation and has been for years, we were confronted again and again by the requirement to assess the relevance of new methods through comparisons to animal studies, rather than through alignment with or fidelity to human biological understanding. And the aim as we see it is not to simply replace traditional animal test methods one-to-one, or even necessarily to produce the same information that is generated by the traditional animal test method, but instead to protect the relevant biological endpoint by demonstrating that the test system is capable of providing human relevant information or information relevant to the species of interest if you're talking about ecotox. So therefore, under the framework, scientific confidence is increased when the new approach captures key aspects of human biology or mechanisms of toxicity, and relevance to human biology can be demonstrated, for example, by incorporating information from human dosimetry modeling, or by evaluating the cell types used in the NAM, or the structure of the target organ or tissue. And given the limitations inherent to animal test methods, establishing the biological relevance of new approaches offers a good alternative for benchmarking its performance instead of directly comparing new approach results to those from traditional animal test methods. And then scientific confidence is also increased when we see concordance between results from new approaches with robust, high-quality human data, which of course would be the ultimate gold standard reference data, but high-quality human data are not always available for every endpoint, as we all well know. So the final essential element, technical characterization, is important to ensure a method is high quality and robust, and it includes a evaluation of the method's accuracy, reproducibility, transferability, the applicability domain, reference chemicals and controls, as well as limits of detection and quantification. And some aspects of technical characterization will be dependent on the method being evaluated and its intended use. For example, increased uncertainty or qualitative results may be acceptable for methods intended for screening purposes, but not for those intended for use in quantitative risk assessment. And this kind of circles back to the importance of fitness for purpose and shows how the assessment of the 5 elements might be iterative. So for technical characterization, it should be demonstrated that the data have been generated using appropriate assays for the question at hand, and the data reporting should allow for evaluation of the new method, including the protocol, the equipment used, and it should also describe any computational methods being used for endpoint prediction or in vitro to in vivo extrapolation. So most of this is pretty self-explanatory and hasn't changed much from the OECD guidance document that I mentioned earlier. So I'm only going to highlight sort of one aspect of technical characterization today, and that's accuracy. So as I mentioned previously, the relevance as well as the predictivity and accuracy have historically been determined by directly comparing results from new approaches to results from traditional in vivo approaches. And our point is that this should not be the default. So the traditional animal test methods are often decades old and were accepted as OECD test guidelines without undergoing a rigorous validation for relevance or reproducibility, as is the standard today. And so, in fact, when we look at the literature, we can see multiple publications describing data analyses which looked at specific endpoints and demonstrated the lack of reproducibility in a number of animal tests. And so you can see here a few examples looking at the variability of rabbit eye and skin irritation tests, skin sensitization in the mouse LNNA, the rodent carcinogenicity bioassays, and acute oral systemic toxicities, among other examples. And as you can see by the authors, several of these analyses were conducted by scientists from Nicetum and ECVAM. The point here is probably not news for this audience, but I think it's important to reiterate that it shouldn't be assumed that traditional animal test methods provide data that's relevant to human biology or mechanisms of toxicity, and they shouldn't be assumed to be providing the quote-unquote right answer to determine if another method is valid. In fact, when you have a test method that can't reproduce itself with more probability than a coin flip, discussion of the test method's relevance to humans is rendered immaterial. So a direct side-by-side comparison of new approach data with data from traditional animal tests may often be inappropriate. Instead, you can demonstrate accuracy by considering consistency across multiple methods, or the new approach's ability to correctly identify positive and negative reference chemicals for the human health endpoint of concern, in addition to greater emphasis on biological relevance and reproducibility, as opposed to quote-unquote accuracy. But I know that as I say this, that currently most regulatory agencies still want to see the comparisons to animal data. And so where possible, and when these comparisons have been made, several factors should be highlighted alongside. So for example, we need to consider which out of the new approach and the animal test is more similar to human biology. We need to look at the reproducibility of the animal test if it's known. And from the publications shown on this slide, we do know that there is quite a bit of variability in many of the animal tests. And the ability of the new approach to predict the results of the traditional animal test method shouldn't be expected to exceed the ability of the animal test to predict itself. And we should note that whatever the reproducibility of the currently accepted animal test, that also represents the currently accepted level of reproducibility by regulators. And so a similar bar should be acceptable for new approaches. Lastly, if a new method is compared to an animal test method, even if the category prediction isn't the same, if you take into account the variability of the animal data, it may be that the error bars around that animal data span multiple hazard categories. So overall, when results from an animal test method and a new approach conflict, the reproducibility and relevance of each method should be considered, and you may find that the differences are explained by the differences observed are explained by the differences in reproducibility and relevance. So to recap, scientific confidence is increased when information about the model and data are publicly available to the greatest extent, reviewed by independent third parties. The purpose of the model is clearly identified, and ideally this is done early in method development. The model has been characterized for its technical capabilities. The model captures key aspects of human biology, or the model shows concordance with human data or across multiple methods. And the use of the framework should enable a confidence building process that allows for the timely uptake of fit for purpose and biologically relevant new approaches that can be used for regulatory decision making. It encourages a holistic assessment of a chemical's ability to cause toxicity in humans, by relying on new approaches that reflect human biological understanding and as such can provide better protection of human health. So we applied these considerations particularly on the technical characterization of a method's accuracy and its relevance to eye irritation in the above publication, which we collaborated on again with the EPA, the JRC and NTP. And we chose eye irritation because multiple in vitro and ex vivo eye irritation corrosion test methods are available as internationally harmonized test guidelines. And yet, despite their demonstrated usefulness to a broad range of substances, they're not routinely used for testing agrochemical formulations. And in fact, the traditional animal test method, which uses rabbits, continues to be requested. So historically, this has been due to a lack of concordance of the in vitro results with results from the rabbit eye test. But the rabbit eye irritation test is notorious for seriously low levels of reproducibility. And on top of lacking reproducibility, there are anatomical differences between rabbit and human eyes and differences in modeling exaggerated exposures in rabbit eyes relative to actual human exposure, as well as subjective qualitative apical outcomes, which may all contribute to this lack of concordance. And ultimately, the regulatory purpose for these tests, whether it's in vivo or in vitro, is protection of human health. So we wanted to assess these testing approaches to see which was most relevant to human biology and mechanisms of human eye irritation. So to assess the relevance and reproducibility of all the available methods, we first reviewed the mechanisms of human eye irritation and the depth of injury model, which, as shown at the bottom of this slide, indicates that there's usually a correlation between the depth of penetration by a chemical into the cornea and the level of irritancy associated with that chemical. Then we considered, once we got a grasp on the mechanisms of eye irritation and the depth of injury model, we considered the mechanisms and the strength and limitations of all the available methods and compared them all against each other. And so I won't go into detail here, but this is a breakdown of the mechanisms of eye irritation, showing the potential, the general potential events that may occur during the development of eye irritation, from molecular initiating events through cellular and organ responses, all the way to organism responses. And so we assessed the measurements of each of the available methods against these mechanisms. As I mentioned, we also took into account the depth of injury model, and we assessed which methods were appropriate for evaluating specific layers of the human cornea. So in the example on this side, we show a section of the human corneal epithelium and the methods capable of assessing effects in the superficial conjunctival or corneal epithelium, the wing cell layer, and then also the lower wing cell and basal cell layers of the epithelium. And details of all of this can be found in our paper at the top of the slide. So once we had assessed all the methods, we found that the in vitro and the ex vivo methods are as or more reflective of human biology and less variable than the currently used rabbit test. We concluded that combining structural and functional information about the test substance with results from human relevant methods will ensure the best protection for humans following accidental eye exposure to agrochemicals. And a major implication from our findings is that the rabbit test does not provide suitable reference data to use to validate new methods, and especially should not be relied on solely to determine the predictivity, accuracy, or relevance of a new method. The other conclusion was that the in vitro and ex vivo methods are ready now for regulatory use for the assessment of agrochemical formulations. So for our next steps for the eye irritation project, we have an upcoming publication on prospective testing where we've performed on agrochemical formulations in a number of in vitro and ex vivo methods. And the publication will go into detail on which methods can be used and how to provide assessment of the full range of severity of eye irritation and predict all four of the EPA categories of irritation, which differ slightly from those of the UNGHS classification and labeling system. And our goal here is that the US EPA could use this publication to put out a formal acceptance of the in vitro and ex vivo methods, though obviously I can't speak for the agency on its planned next steps, so we will see there. And then more generally on the topic of establishing scientific confidence in new methods, we have a couple of upcoming workshops, which we think will be very exciting. We've got some truly wonderful speakers. And one is at SOT in March, and the other is at World Congress for Alternatives in August. So the details for the SOT workshop are at the bottom of the slide, and we're waiting on details for World Congress, but hopefully they'll be on their website soon. And finally, we know that the paper that I mentioned in the first half of the presentation has had a bit of an impact. We know from the EPA NAMS meeting last October that ICVAM is planning an update to its validation framework, and the EPA are in the process of putting one together as well. So if there are any of our projects which you would like more information on, whether I mentioned them today or you heard about them elsewhere, please do feel free to get in contact. And if you happen to be at SOT next month, please come and say hi. So thank you so much for having me, and I'm looking forward to questions and discussion.
Thank you, Anna. Nice. Some of you might be wondering, well, what is the relationship between the two science talks and Anna's talk? And I love the pairing because I think they're very interrelated, the ideas of building confidence and figuring out. And we can see from them some of the questions that we've received about Kelly and Jesse's talks. People are wondering about these topics and how we best can show confidence and transition them from scientific development to application and use. So Anna, let me put one question to you that came in. It's sort of a general question, and Kelly and Jesse, if you want to join the conversation, we've got some questions for all of you in the last few minutes. It's what is a gold standard data set or data? And they're wondering, I don't know if that question is specifically related to the second part of the question, but they're asking about zebrafish as an alternative to model human health and how you measure the concordance between fish and mammals. I know that isn't your field, but I wonder if you have any reactions to that question.
Yeah, so for the first part of the question, what's the gold standard? Well, I think from our perspective, the gold, and I think this is generally accepted as well, the gold standard is high quality, robust human data. But as I mentioned, and yeah, as we all know, this for most endpoints, we just don't have access to it. So then we start thinking, okay, well, what's the next best thing? And traditionally we've fallen back on animal test methods. So that would include invertebrate tests as well, fish tests as well. And I would say that's, if there's existing data from zebrafish or from rodents or primates or dogs, you can use it, but with those caveats that I was mentioning, like you want to know, is this data that I'm assessing against actually relevant? Is it actually reproducible at all? And then sort of take it with a pinch of salt. If you don't get 100% concordance, yeah, you wouldn't expect that anyway, but for any differences that you do see, it could be down to a lack of relevance in the reference data rather than a problem with your method, for example. And I think did that kind of cover the second part of the question as well? I'm not sure.
Oh, I think so. Yeah, thanks. So there's another general question I think that could be answered by Jesse or Kelly. They're wondering who commissions, conducts, and funds these assays. Is it the EPA in this case, a company, or someone else?
I'll jump in. So for the assays that I presented, they're funded by labs at the US EPA. Although there are additional D&T assays that we're hoping to integrate into the battery in the near future that are available in ToxCast that were funded by our European collaborators. But just the data I showed you was funded by the US EPA.
Okay, thanks, Kelly. Jesse?
Yeah, I would echo the same thing. Yeah, ours is funded by the EPA as well. You know, I think there is a lot of intention to, you know, follow the 3 R's, right, when it comes to developing, you know, potential new assays for, you know, for regulatory use. And so there's definitely a large internal interest in funding that type of research. So.
Yeah, and I think it also, I wanted to have that question up because it's absolutely the EPA is on the leading edge of developing many different types of assays and new approaches. But the question of how you then implement them so that, you know, generally a company can use them to assess their products and register them is still maybe an open question that we're wrestling with or thinking about. Jesse, you indicated you wanted to answer one of the questions, so I'll throw that to you real quickly. Do you have a sense of the data gaps, the main data gaps that prevent doing the analysis that you showed for other targets than the ones that you looked at?
Yeah. Thanks to Amy for the question. She also is a postbac at EPA, and I figured it might be, you know, kind of translatable, you know, to Anna's work as well. And there are definitely a lot of gaps, right? To kind of expand or, you know, really understand what is the full domain of applicability of an assay like transcriptomics. You know, this analysis that I presented is really based on having robust information about, you know, certain reference chemicals, and so we're essentially relating everything to a group of chemicals for which, you know, we may have some in vitro data, we may have some in vivo data, and it may have some level of uncertainty associated with it. So I think there are a lot of opportunities to kind of compile more robust sets of these chemicals. That's obviously going to require additional testing on the in vitro side to establish confidence in that particular response along an adverse outcome pathway, you know, if you choose to do so. So yeah, I think it's, you know, this is kind of a proof of concept, but I think it's going to require a lot more thought into developing chemical sets like these, and then also kind of distilling down and saying what is the actual NAM that we would like to proceed with to assess this particular mode of toxicity. So hopefully that answers her question.
Yeah, I think there's some great sort of bringing it out to a more general point as well. I'm afraid we have run out of time. I was going to try to squeeze a couple more questions in, but thank you all so much for your talks, and thank you for answering all of these questions. I know you were busy and behind the scenes answering some of them by typing, so thanks very much. Thanks all of you for joining us. As I said before, the recording will be posted on the website, and hope you'll join us next month again. And thanks again to all the speakers. We are very much appreciated. There are lots of really nice comments in the chat. All right, everyone have a great day. Thank you.
Thank you.
Thank you.